Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Teknologi mampu menghapus batasan jarak, waktu, bahkan ukuran. Seperti bagaimana saat murid saya mensimulasikan materi sintesis protein dalam misi menyembuhkan pasien kanker. And now, witness how my student used MetaQuest 3 to simulate it. Here we go.
Hello friends, today we're going to be using VR to learn about biology. Now today we have a certain mission, and this mission is to cure a cancer patient. Now, how do we do it, and how are we going to cure this person is by learning about our cell and going through several processes throughout the way. Now, first of all, I'm going to click on the application right here. Okay. Now we're in. Okay. And okay, let's speak English. Okay. Okay. So this is the intro menu. Okay. So within this entire process of healing this person from cancer, we're going to be doing it by helping the cell make a certain protein in order to be synthesized and sent to the correct target in order to heal this patient. Now, there's going to be three processes right here. We have the preparation room, transcription, and the translation. Okay, so we're going to start off by going to the preparation room. Okay, let's go. Let's see. Mine's getting hard. Okay. Now, before we help the cell cure our patient by protein synthesizing, we must first acknowledge and use our learning process result in order to identify each part of this cell right here. So I'm going to start off by putting, let's see, nucleus. Okay, let's see, nucleus right here. Oh my god. Okay, this nucleus, I'm going to put it right, okay, let's see, where's nucleus? This is going to be our nucleus because this is the center of the cell. This is the entire the genetics, the DNA sequence. This is where it is. Okay, let's see. Okay, we got the nucleus. Now let's see what else we got. We have also nuclear envelope right here, which is envelope is something that it acts like something that covers it. So since this is a nuclear envelope, so it must cover the nucleus right here. Yes, that's it. And let's see. Next, I'm gonna go to the mitochondrion. So mitochondrion, I think it's gonna be right here, since this is like a circle-shaped thing. I don't know what that is, but that's certainly the mitochondrion. Next thing, we're gonna go with the plasma membrane. This is gonna be right here, since plasma membrane is the part of the cell that covers the cell, protects the cell, so it's gonna be this part. Next thing is the rough endoplasmic reticulum, which we know the rough RE is the RE that is covered by ribosome, so it's gonna be this part, which is near the nucleus. Next thing is the Golgi complex. So this blue part right here is the Golgi complex, and it's gonna be related to our process of protein synthesis later on. So I'm gonna put it right here, and we got two left right here. Let's go with the ribosome. Ribosome is obviously, as I explained earlier, it's this tiny red shaped thing. So that's gonna be this one. And then the cytoplasm. Cytoplasm is kind of like the substance that is all over the cell, so it acts as a medium in order for the entire process within the cell to actually work. So there has to be a medium. So cytoplasm is that medium. Okay, let's put it right there. Okay, voila, we done the process. Now we're gonna move on to the next one. Next step. Now watch me teleport. Bam. Okay. We're gonna take this thing, teleport again. Bam. Let's see, we're gonna put this thing right here. Right there, okay. Bam. Now we're moving on to the next step. Let's see what we got next. This entire thing is actually fun for me because rather than just learning it through books or through materials, I get to practice it. So here we have a DNA sequence. Now this process right here is what we call as transcription. Is where we put in the mRNA sequence in order to later on be translated into the certain protein that we're going to be making. So let's see right here. We have guanine. Okay, so guanine is in pair with cytosine, and thymine is in pair with adenine, and this. Also a little bit of facts. So mRNA doesn't have this thing called thymine, so it's changed into uracil. So when we see A, it should have been thymine, but since thymine is changed to uracil, we're going to be putting U right here. And then C, we put that G again. We put C again, A, so we put U again, thymine, so we put A again, okay, we get A, so uracil, wait, and this thing again, and lastly, is this the last? I don't know actually. Okay, so we have completed the process of transcripting the mRNA, okay. So after this process right here, the mRNA is going to be transported to the ribosome, in that process we're going to have the translation, in which we're going to make the amino acid. Okay, so where do I go? Okay, I'm going to be teleporting. Okay, how do I, wait. Okay, so we have this thing, we're going to be putting it right here, okay. Ooh. Let's see. Voila. Now we're going to be moving on to the translation phase. Okay, so we're here in our ribosome. Okay, so where is it? Okay. Okay. Now we're going to be continuing the process of translating the mRNA, the tRNA. Now it's going to be becoming the amino acid. So let's see right here. We have ACU. That's going to be the THR. Okay, let's pick this thing up and let's put the corresponding sequence codons. Okay, let's see. So we got A, that's gonna be with U. Okay. Wait. Okay. Okay, this right here. U, and we're gonna ask C, that's gonna be with G, and the last one is U, so that's gonna go with A. And then let's put the THR. Where is it? Okay, this. Now, let's see. Okay, next one we got the UGU codon, so that's gonna be the cysteine. So the THR is actually threonine, and next one we got cysteine. Okay, let's see UGU. We're gonna put in A, C, and A again. Okay, we're gonna be putting cysteine. So where's our cysteine? Okay, this is it. There we go. Then Bam. Okay. Next one we got GCA. That's gonna be alanine. Alanine. So let's see. We got G. The sequence gotta be G. Wait. G. We got C. We got G. And lastly we got A. That's gotta be U. Wait. Okay. So alanine. Where's our alanine? Okay, right here. Alanine. Okay. Put it there, buddy. Okay. We got the AUG. This codon AUG. It's gonna be methionine. So we're gonna pick this. Okay. Let's do this first. That's AUG is gonna be UA. Wait. No. And methionine. Okay. This is it. Okay. Now we've completed the translation process. Now we have the entire sequence complete, and the ribosome is actually synthesizing the protein right now. So we have this sequence right here. Beautiful. Now we're gonna be going there, right here, and this process is called modification. It's before the desired amino acid, desired protein is transported to the target of its protein. We have to first modify it, and this is what the Golgi complex actually do. It modifies the desired protein by adding carbohydrates and such. Okay, last process. Okay, we've got it done now, and then we're gonna put it right here, and it should now be transported to the desired target cell. Okay, so I think we've pretty much done the job now. Okay, so thank you so much. Now we have cured this cancer patient by helping our cell do the job by going through several processes, and I hope you guys learned something from what I did earlier. And I think that's all. Assalamualaikum warahmatullahi wabarakatuh.